Hi guys, so this is a short video kind of summarizing and bringing together some of the key things that we need to know about the structure of DNA. And again, this information is going to help us to see sort of how this molecule functions, stores information, and actually gives us sort of some of the physical traits we see. So how can we use this to understand why we have the hair color we have, or the eye color, or the blood type? And so we'll see kind of how does the structure of the molecule at that most foundational sense allow for the encoding of all of that information that makes us us. There's just five things we're going to kind of touch on. We're going to do a little bit of history, and then we're going to talk a little bit about the structure of the nucleotide, so the building block of our DNA molecule, as well as complementary base pairing. We'll talk about some of the bonds that we find within this molecule, naming them and talking about their significance. We're going to go back to this word anti-parallel, which I know is kind of new, and talk about what it is, make sure we have a good understanding there, and then understand why this is important for understanding. So kind of a preview of coming attractions when it comes to DNA replication. And last but not least, this is really just my attempt to sort of hammer this one home, is looking at how does this structure actually do its job? So how does it encode that information for actually making us us, for making the distinct proteins that give us all our unique physical appearance? So history. In terms of history piece, so we're not a history class. This is more just kind of a social science piece, because the story of the discovery of the structure of DNA exists so much in the zeitgeist of our society, and you'll hear people name drop these people all the time, that it's something that, I feel like we just want to know. Even if you just hear it once, then later on someone makes a reference and you're like, oh, I understand that reference. I know where that came from. So to understand kind of the structure, or the history behind the structure of DNA and its discovery, we actually have to go back to kind of the 1860s, so quite a far back in history, and look at a man by the name of Friedrich Michener. Now, I know his name is not up on here, so this is a quick one. Michener was actually a Swiss, I think he was a physiological chemist, and his lab was set up right next to kind of a field hospital. So he used to go over to this field hospital, and he would actually, this is super gross, he would take kind of the pussy bandages from the soldiers that were inside this hospital, and he was trying to figure out kind of what is this pus, so what is this white substance. And so he would do a variety of kind of experiments on them, try and treat it a different way. And he actually was the first guy that actually ended up isolating DNA in its pure form. Now, if you go back to Michener's day, though, people really wouldn't have cared that much about DNA, because at the time, people believed that what makes our unique characteristics and all of the information that is passed on from parent to child and all of that stuff was in proteins. So people really didn't care that much about DNA at this point. Now I have to go all the way back to fast forward a long time, and we're around 1919, where a man by the name of Levine publishes sort of this seminal work on DNA. Now at this point, people have figured out, no, DNA is important. Like, this is the molecule that actually makes us us. And Levine does a bunch of experiments and figures out kind of what makes up this molecule. And so kind of preview of what we'll see, Levine comes out and says there's a phosphate group in here. There's these nitrogenous bases. Here's the elemental composition. And so he publishes these papers. He's like, no idea what they look like when they're combined, but here's what's in there. We then get to the 1940s, and this one you guys have heard of before, and we get Edwin Chargaff. And Chargaff figures out that, yeah, there's nitrogenous bases in there. So there's adenine, thymine, guanine, and cytosine. But I find that when I look at DNA, A always equals T, so there's always equal amounts of adenine and thymine, and there's always equal amounts of guanine and cytosine. And so that was kind of the work that had done when we get to the point or the beginning of our story here. So looking at the 1950s. Now, at the time, there's always in science kind of the next greatest and latest discovery that labs are competing for. And at the time in the 50s, it was the structure of DNA. People had read Levine's work, they'd read Chargaff's work, but no one had kind of a model of this molecule that worked. So that would actually be stable, where it had all the bonds, molecules, elemental composition, followed Chargaff's rule. And so you had all these labs around the world kind of competing to come up with that next great model. Watson and Crick at this point are kind of no-name scientists. They don't have any big published work, and they're out of King's College. But they are friends, and they're mostly model builders. And they're really good friends with this guy by the name of Maurice Wilkins, who has a pretty prominent lab there at King's College in England as well. What happens in the 1950s is that Rosalind Franklin, who is a postdoc student who had been living in France at this point, and she specialized in looking at protein structure by doing what's known as X-ray crystallography, meaning she would literally take X-rays of these tiny molecules and kind of look at what the shadows looked like and the structures, and try and take lots and lots of pictures to figure out kind of what does the shape look like. And so that's how she was figuring out the shapes of molecules. But again, she's focused on proteins. She's not in the DNA land. Watson and Crick are trying their best to try and figure out what this structure is, and they've been talking to Wilkins a lot. They go out, they hang out. And Wilkins has a pretty contentious relationship with Franklin. He is pretty sexist and doesn't think that, a woman should be in the lab. And when she shows up, he tries to treat her like his lab assistant. So they have a pretty terrible relationship between the two of them. And as the story goes, one day Wilkins goes into Franklin's lab and he sees this picture right here on her desk. And he sees that X-shaped structure. And from talking to, and that she has it labeled as DNA, and he had been talking to Watson and Crick, and he knew that he was looking at something that would help them to figure out what this structure looks like. So he takes the picture from his desk and gives it to Watson and Crick without Franklin's approval. So essentially stealing some of her work, giving it to someone else. Watson and Crick obviously look at this picture and realize that what she's essentially taken a picture of is sort of the top-down view of the double helix of DNA. So those X's are actually created by that twisting shape of the DNA. And so they come up with what's known as the double helix model. They publish it in Nature, and they credit themselves, so Watson, Crick, and they credit Wilkins with it as well, and they do not credit Franklin with it. They ultimately do kind of talk about sort of her role in the picture and stuff, but they downplay it greatly. And when they go on later to win the Nobel Prize for discovering the structure of DNA, by that point, Franklin has died, and they do not award Nobel Prizes posthumously. So she's kind of historically known as sort of the woman who got no credit and sort of had her work taken from her, which is kind of a terrible story. But that's the kind of a important story and that you'll hear when it comes to the structure of DNA. But what we end up with is we end up with this model right here. So again, the double helix model that Watson and Crick came up with, it has not changed since the 50s. There's been no evidence to suggest that it's incorrect in any way. And again, this model is called the double helix. Double because there's two sides, and helix because there's a twist in the molecule. Now, within that much larger molecule, we do have these smaller molecules, these building blocks called nucleotides. And so we're going to look at what actually makes up those nucleotides. So right here, you guys have seen these. They were in part of your worksheet you did last time. And there's three major parts of a nucleotide. I've labeled them 1, 2, and 3 here. So see if you can't remember what some of these parts are. Good, so hopefully you're able to identify that 1 is your phosphate group, 2 is your 5-carbon sugar, and 3 is your nitrogenous base. So again, this is the basic building block of DNA, and these are just the 3 components of that building block. One of the patterns that we'll see when we look at really large molecules inside living things is that they tend to have sort of this brick, this base shape that just repeats over and over again. What we're looking at right here is that base shape in DNA. We're looking at that building block. Now, there's a couple different ways that you're going to see this. In really simplified models, they might just represent the structure of these or the combination of atoms that make up these 3 components as shapes. We had gone through this and talked about this last week, and I just want to drive this point home. When you look at a circle attached to a pentagon, attached to a rectangle like this, this is just a basic visual representation of the arrangements of atoms that give us each of these components within our molecule of our nucleotide. So this circle right here is supposed to represent the phosphate atom as it's attached to the 3 to 4 oxygen atoms in this hydrogen. So regardless of whether you see it in its structural formula, so right here with the nitrogens and oxygens and the carbons, remember those, if they're invisible but there's a bend, it's a carbon. So the carbon's in here, or you see it sort of in its most cartoony-ish form, looking just like a bunch of shapes, they're both supposed to be representations of that building block, so of that basic shape. Now, when it comes to the structure of these nucleotides, it actually has 2 different functions. So the sugar and phosphate is a constant component within our nucleotides. So if you look right here, you're looking at half of a DNA molecule. So this is like if someone took the double helix and undoubled it, so peeled the 2 sides apart. So if you look at one side here, you can see your nucleotides, and you can see kind of that joining we talk about where it's just repeats of nucleotide attached to one another. The key component here is that the sugar and phosphate are constants. So if you look all the way down the side of this molecule, every single phosphate looks like every other one. Every single sugar looks like every other one. And these are the structural components of a DNA molecule. A DNA molecule literally encodes the information for a cell on how to function, which means that you need this molecule to be stable. You need it to not be something that is easily going to break or be damaged, or else we would have a whole lot of problems being multicellular organisms. And so what we find is that that structure is provided by those sugars and phosphates connecting to one another on the outside of our molecule. And I'll show this to you guys in a much larger model in a second. The second function of our nucleotides is they encode information. So if you look towards the middle of here, you'll actually see your nitrogenous bases. So you'll see adenine looks different than guanine, looks different than cytosine, looks different than thymine. There's no label on this one. This is thymine. And so each of these kind of variable parts right here, based on the sequence within a DNA molecule, is going to give us our unique fingerprint. So no two of us that we know in the world, except identical twins, which we'll talk about a little bit later in genetics, have their adenines, guanines, cytosines, and thymines in the exact same order. And that's what gives us our inherent genetic variation. One of the key features that we'll notice about DNA is that it has a uniform width. And that has to do primarily with the fact that your nitrogenous bases actually have different structures. So this was in your video. So some of your nitrogenous bases have 2 rings, and they are known as purines. So in DNA, that's adenine and guanine. And then you also have some nitrogenous bases that are just 1 ring, and those are known as your pyrimidines. Cytosine and thymine are found in DNA, and uracil is kind of your new friend here that you're seeing, is actually found in RNA, so ribonucleic acid, which has a slightly different structure than DNA and will be a huge focus for us when we get into protein synthesis in a couple units. When we look at the double-stranded nature of DNA, what we find is looking down the middle of the molecule. So here's your here's your phosphate, your sugar, and then here's your nitrogenous base. So we're looking at a much more abstract sort of depiction here where it's primarily shapes that are used to represent those nucleotides. And again, this dashed line here is a nucleotide. We notice that whenever you have a cytosine on one side, the opposite side, so the other side of the DNA molecule, has to have a guanine attached to there. And this has to do with sort of the physical configuration of these molecules and the number of bonds they need in order to be stable. So when we look at cytosine and guanine, it requires 3 bonds between these two in order to get stability, whereas if we look at adenine and thymine, it only requires 2 bonds. What happens if you were to try and bond adenine with guanine, they would form 2 hydrogen bonds, but then you would have part of your guanine molecule that would be unstable because it requires an additional bond in order for it to be sort of happy, for it to be a stable molecule. So that's why it's always going to be cytosine with guanine, always adenine and thymine. And that's what you want to make sure you always remember about your complementary base pairing rules. Again, because you have guanine and you have your adenine as being double ringed and your thymine and cytosine is single ringed, and it's always A with T, G with C, what you actually find is that there's a uniform width within this DNA molecule based on the fact that you always have kind of a net 3 rings across your molecule. So it's always something with 2 rings bonding with something that's 1 ring. This is kind of my goofy memory trick when it comes to purines and pyrimidines. If this doesn't work for you, obviously you're always more than welcome to come up with your own. So I always remember pure Ag. So Ag is the elemental symbol for silver on the periodic table. And so that reminds me kind of that purines are going to be your adenine and guanine. And these are kind of a little bit ironic because they're purines, but they actually are the more complicated ones with the double rings. And my second memory trick was I always remember cut pi, C-U-T, all capitalized, because cytosine, uracil, and thymine are your pyrimidines. And these ones are going to be, you cut a pi in half because it's only 1 ring. So it's literally the halves of your purines. Those are my goofy memory tricks. If they help you, you are more than welcome to borrow them and use them. All right, so let's check ourselves really quickly. So if we have right here, so if one side of my DNA strand, if the sequence of nitrogenous bases is AGTACG, what has to be on the other side? All right, so hopefully you're able to get it. It's TCATGC. We'll do one more of these. How about this? If you had this on one side, so TTGATC, what you have on the other side? Good, so hopefully you got this one, AACTAG. So again, that's that complementary pairing. So this is kind of when you see a sequence like this is like the most shorthand version of the structure of DNA because it's basically ignoring phosphates and sugars and just being like, yeah, we know we're there. But this is what you would have for your nitrogenous bases down the middle. When we look at bonding in DNA, there's 2 bonds that we primarily want to know. Oh, my underlines are still here. So if you look, you have kind of one type of bond right here that it's going to be represented by a solid line and will be a very strong connection between molecules. Do you remember what type of bond that is? Good. So hopefully you remember, took me a couple clicks to get there, that any solid line you see in one of these diagrams represents a covalent bond. And covalent bonds are very, very strong. Now you're going to find covalent bonds kind of two different places in your DNA molecule. The first one, you'll notice that I have this kind of purple outline right here around one of my nucleotides. And you'll notice you've got a solid line connecting your phosphate to your sugar and another solid line connecting that sugar to your nitrogenous base. And that is because it is strong covalent bonds that hold a nucleotide, so a building block of DNA together, which is good. We want those building blocks to be solid. We want to be able to add them, stack them up, build larger molecules. You also notice, remember, if we go back to kind of when we talked about the basic components of a nucleotide, we said that this sugar phosphate connection that makes up the outside of my DNA molecule also needs to be strong. This is what gives our molecule its stability, means sure that it's less likely to get mutations, damage, still be able to kind of, we'll make sure that it can hold all that information we need it to. So what we find is that when you add nucleotides together, so when you join one building block to another, you form a covalent bond between the phosphate of one and the sugar of another. And that new bond that would form as we add these nucleotides is what I've circled for you guys in green right there. Now, this has a special name. It is called a phosphodiesterase bond based on kind of where it forms in the molecule. I'm not going to make you guys responsible for that, but just in case you ever come across that word, don't let it throw you off. It's just the special name for the covalent bond that attaches one nucleotide to another in order to build those large molecules. The second bond that we want to pay attention to are these dashed lines that I'm seeing between my nitrogenous bases. What type of bond is that? Okay, so hopefully you remember those are nitrogenous bases are going to be connected by much weaker hydrogen bonds. They're actually kind of a misnomer. Hydrogen bonds are really weak association between atoms that have partial charges. So in this case, it would be a hydrogen that's acting like it's positive and then an oxygen that's acting like it's negative. And because they're oppositely charged, they have this weak attraction to one another. And so what we get is kind of what holds the two strands together. So one side of a DNA molecule to another is just a series of these weak associative bonds called hydrogen bonds. Now, this might sound bad initially, like you're like, oh, Ms. Curley, structure is so important. We need a strong molecule so it doesn't degrade. We need to keep that genetic code sound. But we're going to see that when we go to look at replication, so how DNA actually copies itself, that one of the things we need to do is peel these two sides apart. So we're actually going to separate the two sides of our DNA when we copy it. Having weak hydrogen bonds, attaching them is actually helpful because it makes it much easier to kind of go through and break these connections in order to create those new copies that we're going to. So quick ways for how you'll see kind of this larger molecule represented. So you might see, this is kind of the most accurate representation of the actual structure of DNA. So where you see the double helix with all the twists in it. Most commonly when you're looking at the structure, they'll kind of unwind it for you. So it'll look more like a ladder or look like kind of the diagram we were just working with. And you just have to realize this is an untwisted representation of a DNA molecule. So they're assuming you know, hey, we've kind of unwound it to make it easier to read. If you ever come across one of these ladder diagrams, so this is the one in the middle, you'll notice that they've kind of ignored the sugar and phosphate. They're not even diagramming them out as separate molecules. They're literally just calling that backbone structure because all it is that structural support to keep this molecule together, keep it sound, as just a solid block. And so keep in mind, if you ever see that solid piece right there represents that sugar phosphate along the sides or outside of the molecule. All right, number 4, get into what it means to be anti-parallel. So anti-parallel is a vocab term that means that the two sides of your DNA run in opposite directions. Now, this seems kind of like a strange thing to say, but if you actually look at kind of your DNA molecules themselves, let's pay attention to the sugars because they're the easiest ones to look at. And you'll notice that you kind of have a pentagon shape with a little point on top right here. And it's not drawn in this diagram, which is kind of a shame, but that's actually an oxygen atom right there that sits on the very tippity top point. And you can treat that little pointed part of your pentagon sugar as an arrow. And so if you follow the arrows, you'll notice that one side of your DNA molecule points up, and you'll notice the other side of your DNA molecule points down, meaning it literally means the opposite side is kind of in a total opposite orientation. So they are sort of opposite, flipped to one another. There's actually a naming convention that goes with this anti-parallel, and that is 3' and 5'. And so you will hear when we get into DNA replication, I will say things like DNA only replicates 3' to 5', or your nucleotide attaches to the 3' sugar. What we need to kind of remember here is I'm going to show you guys where this naming convention comes from, just so when you hear that 3' 5', we have a foundation to connect that to. So you'll notice that in the bottom center, you're looking again at a structural formula of a nucleotide. The only thing that's not shown to you is your variable base. So this could be adenine, guanine, cytosine. But again, this is not a structural component on the outside. It's not going to be involved in labeling 3' 5'. Remember, that's just an encoding portion of the molecule. So it's okay that it's just labeled as base. And we're going to focus back in on that 5-carbon sugar. Now we remember that in these structural formulas, each of these bends represents a carbon atom. Now, if you ever take organic chemistry, you will learn sort of why these are numbered in the sequence that they're in. So why does this one right here get to be number 1 and this one's 2 and this one's 3? It has to do kind of with rules about the orders of importance for functional groups that are attached to each carbon. We are absolutely not going to get into these. The only thing I want to show you in this diagram is that this carbon right here is labeled 1, this one's 2, this one's 3, 4, and then 5. When we call the orientation 5', you're essentially looking for that phosphate group, because that phosphate group is going to be attached to that 5' sugar. So if you look at how I've labeled this diagram over here, the easiest ones to fill in are my 5'. And that's because I look right here and I notice that my phosphate group is pointing up this way, so I know that's my 5'. And then I look down here and I notice what's pointing down on this end. Oh, it's this carbon right here, so my 3' carbon. So this side, so I'm looking over here on my left-hand side, is oriented from 3' to 5'. Now, because I know they're anti-parallel, I should know, hey, well, if this one runs 3' to 5', well, then the other side has got to run 5' to 3'. But we'll check ourselves anyway. So if I look over on my right top-hand side, I notice what is pointing up. Oh, it's that 3' sugar on the bottom of my nucleotide. And then I look down here and, oh, yep, there's my phosphate group pointing up. So that's my 5' carbon that's pointing up this way. So this side does run kind of 5 to 3. So besides being super awesome and knowing kind of the fun fact that DNA is anti-parallel, and that means that essentially the carbons are oriented in opposite directions, and we can label that opposite orientation 5' to 3' and 3' to 5'. Let's get into why that matters so much. So I am not just trying to give you some DNA fun facts or make your life tough. When DNA goes to replicate, the only place new nucleotides can be added, so the only place you can make new connections between nucleotides is at the 3' carbon. So you'll see right here in this diagram, you kind of have a basic preview of coming attractions, and you'll see right here, see how when you're building this new complementary strand, which is shown in the turquoise, off of your original purple strand on the bottom, you can only add those new nucleotides when that 3' spot is open. Now we're going to see how when we do DNA replication, that means that one side of my DNA molecule is going to have no problem making a copy of itself, because that 3' carbon is going to be exposed when it splits, so it will literally just make a new strand in a single file. We're going to notice the other side of my DNA molecule has an issue, because when you split it, it's the 5' carbon, or the one that has the phosphate group, that ends up being exposed, and you can't add new nucleotides to that one. The way that I always kind of think about this is I think of your nucleotides as almost like Lego bricks. So here you have kind of the side view of a Lego brick, and Lego bricks really have to be ordered and oriented in a special way in order to get them to join. So for example, if I was trying to connect these two bricks together, you'd be like, Miss Curley, you're crazy. There's no way those two things are going to get stuck together. And this is kind of what happens when you have that 5' carbon exposed. There's just no place for a new bond to create in order to add new nucleotides. However, when you have that 3' carbon exposed, it's like you have it in the right orientation. They're arranged so when you bring them together, like my two Legos here, they're going to join together. So it just has to do kind of with the availability and the orientation of the molecule. All right. Last thing we want to know about DNA, and again, this is just kind of a bringing it all together, hitting that kind of, what's the point of DNA? Why do we care about the structure? So DNA is the molecule that encodes all of the instructions to make you, you. What that means is that essentially you as an organism and all living things on Earth are a series of recipes for different proteins. And we all make slightly different proteins, and that gives us our slightly different appearance and our slightly different physiological functions. So I have a different blood type maybe than you. I have a different hair color than you. That literally has to do with the proteins my body is making. how my body knows to make those proteins is it literally is reading the sequence of nitrogenous bases. So the A's, T's, G's, and C's that are inside that DNA molecule. So the cool thing here we want to kind of appreciate in this last moment in this section is that you have this large DNA molecule made up of that phosphate, sugar, and nitrogenous base. That phosphate and sugar is forming that structural backbone to make a strong, stable molecule. And those nitrogenous bases down the middle, that sequence and that like that you have of each of those nitrogenous bases is encoding or storing the information that creates your unique genetic fingerprint. So literally kind of your unique identity.